The overall goal of these skeleton and fractal analysis methods is to measure microglia morphology from IHC-prepared tissue, using quantitative methods that are both high throughput in nature and sensitive to detect small differences in cell shapes.
This method can help answer key questions in the neuroinflammatory field by defining the nuances of microglia form and function during health and disease. The main advantage of this technique is that it quantifies microglia morphology using continuous rather than categorical variables. The methods described here do not require proprietary software, and it may be used for screening of brain regions or cell-by-cell analysis. Demonstrating the procedure will be Kimberly Young, a technician from my laboratory.
Before beginning, ensure that the Analyze Skeleton plugin has been downloaded to ImageJ or Fiji. Then open a 30-micron Z-stack showing IBA1 immunostained brain. If using a fluorescence photomicrograph, ensure the image is 8-bit and convert to grayscale to best visualize all positive staining. Use the toolbar and click Image, look up Tables, Grays. If using a DAB brightfield photograph, first use the FFT bandpass filter plugin with the default settings. Click Process, FFT, bandpass filter, and then convert to grayscale. If the image is too dim to visualize the process of the microglia, use the toolbar and click Image, Adjust, Brightness Contrast. Adjust the minimum or maximum sliders as needed, up to the edges of the histogram, but no further.